Gee, I could listen to Lee all night. It's a real honor to be introduced by Lee as a longtime friend and colleague, and I think at least equal iconoclast. And it's truly an honor to have you here opening this tonight. And Lee's played a very critical role in my research, and when we get to that point, I will point that out. He changed the world for me and helped me change it for others. So he's one of the most important American scientists ever. So I'm going to tell you about life at the speed of light, which is really about the interchange between the DNA world, the chemical world, and the digital world, and show you how, from the work that Lee started with the first automated sequencer, we could read the genetic code, digitize life, and fill our computer databases with this digital biological information. And then we decided to actually prove that DNA was in fact the key thing carrying all the information for life. We started at the other end. We started with the ones and zeros, remade the DNA chemically, and booted it up to create new life forms. So I'm going to take you through that journey. And one of the things that I found in doing this is, you think you have a new idea, and then you do a little research and find out somebody wrote a book in 1901 on synthetic life. In fact, was so sure that somebody would someday make synthetic cells that this Frenchman argued nobody should be surprised when it happens. So the surprise in science is we very quickly lose our historical perspective. And the exciting thing about this field is everything has happened in the span of a single lifetime. It's happened, Lee's older than me, so it happened totally within his lifetime. You have to add a few years on to have it happen within mine. But the place that I argue that it really started was with a physicist, Schrödinger, in 1944. And Schrödinger gave a series of lectures at Trinity College in Dublin titled, "What is Life?" And he was basically asking, does life obey the physical principles, or is there something special about life that's different? And in the process of doing this, he came up with some notions about what he called the code script. Ended up being predicting much of what happened in the later discoveries. And I think that's the main reason we remember him. But he wrote a book based on his lectures, and this is the book. It was a very thin book, but I think every modern scientist that's made significant contributions in biology has been inspired in part by this book. Watson and Crick went at length trying to sort out what the code script was, and after they determined what it was, Crick actually called Schrödinger and told him that his early descriptions ended up being, in fact, very accurate. So the problem was in the 1944 when this was put together, and Schrödinger predicted the code could be as simple as the Morse code. All biologists apparently thought that DNA was much too simple of a molecule to carry all the inheritance for the entire biological world. And so they were sure that it was proteins. And this is one of the dangers in science. Just like any other field, belief systems can set people back and set back knowledge for decades. And because this absolute certainty that proteins were the genetic material, people ignored data even when new data came along. So the world started to change, at least in retrospect, also in 1944, when Oswald Avery and his colleagues did a simple but very elegant experiment. They basically used a series of enzymes that destroy DNA, RNA, and protein sequentially and proved that DNA itself was the key information-carrying molecule. their data went largely or totally ignored for quite a while. This area got a boost when a few years later, Sanger determined the first protein sequence. And until this was done in showing that proteins were actually linear molecules of sequential amino acids, the theories were that these were sort of loose aggregates of substances and not a clear linear code. And then the big breakthrough came with Watson and Crick's model to show what ended up being the structure of the double helix of DNA, showing how the DNA molecule itself could be self-replicating and drive copies of itself in a very nice fashion. But even when they published their paper in 1953, there wasn't an immediate recognition that this was a major breakthrough. Several more transformation studies were done, and the field slowly started to change. But I think it wasn't probably until the early 60s when Marshall Nuremberg and Gobran Khorana actually discovered this triplet code of letters in the genetic code that code for amino acids. And I think at this time, people began to really recognize in a solid fashion that DNA was indeed the genetic material. One of the first molecule sequence was done by Bob Hawley. He actually sequenced the transfer RNA molecule, and this is key because this is the link between DNA and proteins. It carries the amino acids into the ribosomes. The 70s saw some additional changes. My friend and long-term colleague Ham Smith published the first paper discovering restriction enzymes. These are the molecular scissors that cut DNA in very precise points. And we basically wouldn't have a molecular biology industry if it wasn't for those enzymes, because they allowed cutting and splicing of DNA. And I argued that the 70s brought some real breakthroughs along with some very bizarre dress styles and fashion. But Paul Berg and Cohen and Boyer did some of the first actual gene splicing experiments. Stanford patented their methodology, and this led to actually development of human insulin as the first recombinant drug with Genentech and Eli Lilly. It wasn't until the mid-70s that the first genome was sequenced, and that was actually an RNA virus. A year later, Phi-X174 was sequenced by Fred Sanger and his colleagues, and this new sequencing method, now referred to as Sanger sequencing, led to Sanger getting his second Nobel Prize, and then led to this expansion that we could all do with DNA sequencing. But even with that, it took another 18 years before, as Lee described, we could sequence the first genome of a living organism. And this was based on the key discovery that Lee's lab made of showing that, in anybody that sequenced DNA with these early techniques, they were really torture to do. You had to use large amounts of radioactivity. It was more an art form than a science technique. And what Lee showed you is you could get rid of the radioactivity, and he put 4 colored dyes on the different letters of the genetic code, and they could be activated by a laser and read out sequentially into a computer. I actually got one of the first machines that was based on this technology, and that started the revolution that led to us being able to do the first genome. So without the new techniques and without the automated sequencer, genomics would not have happened at all. So Lee, thank you constantly for that. 5 years later, we went from 1.8 million letters of genetic code to 3 billion for the haploid human genome. And things have continued to scale dramatically from there. We're filling our databases with these ones and zeros, and the challenge for understanding that information is huge. In parallel to all this work on DNA was some continued work on proteins, but we started to really understand how we could go from this linear code of 4 letters to the ones and zeros in the computer, and continued digitization of biology. But it became, as we got more and more data on protein structure and function. And I thought this book by Charles Tanford and Jacqueline Reynolds best described the protein world as nature's robots. And basically what these are all the proteins that carry out and form basically the function that we see and feel with life. And they're based on the linear code of DNA coding for the linear code of proteins. But within that linear code is everything else that's needed for the function. So I like describing life as DNA-driven cellular machines. And each of our cells contain hundreds to thousands of DNA, of protein and RNA robots, read and formed continuously from this DNA software. And so hopefully you'll see in a few minutes that every bit of what happens in life comes from this linear DNA code. We have multiple protein types. We have things that elastins that form our arteries, and they stretch like rubber, to structural proteins, to everything you can imagine about our physical structure comes from this protein world. There's some beautiful structures, and these are not simple robots. And this came from 2005. This is the complete three-dimensional structure of the bacterial ribosome. And this is comprised of multiple protein units, multiple RNA units. And this is the key machine that takes the RNA message, reads that linear code, and turns it into linear proteins. Without this machine, there would be no life. So the machinery inside the cell is essential for carrying out the instructions in the DNA. But everything in this beautiful structure came from the linear DNA code. Protein folding has been sort of an enigma to science for a long time because it's a huge problem. If you have a protein just with 100 amino acids, there's 2 to the 100th to 10 to the 100th power possible folding conformations. So if you had to try each and every one of those, it could take tens of billions of years to try them all. But built into this linear protein code that comes from the linear DNA code is all the folding instructions for this to happen in a few microseconds. But probably the thing that surprised me the most looking into all these processes is that it's driven by Brownian motion. And anybody who's ever looked under a microscope understands what Brownian motion is. It's that jiggling that you see of everything under a microscope. And when this was first discovered by Robert Brown in 1827, he thought he had discovered the absolute secret of life. But it wasn't until 75 years later that Einstein actually showed what he was looking at under the microscope, and it's the water molecules in constant movement driving energy processes. So we always show pictures of DNA and proteins as static entities. But if you could really see them, they're jiggling and moving around. The equivalent inside a cell is like the equivalent of a Richter 9 earthquake, constantly moving. Machinery is built sort of like this last bullet, like a bicycle that can't go backwards, so the energy drives it forward. And lots of our molecular machines work in that fashion. Here's actually a movie showing Brownian motion simply by putting in some fluorescent nanoparticles in. So if you were looking at the molecular scale inside any cell, this is what you would see, only you'd actually see the proteins as well moving around at this high speed rate. So all this is driven by a temperature, which is one of the reasons life can only work in a certain temperature range. Protein folding, I'm going to show you a movie of this over seconds, what takes place in 6 microseconds. And you'll see the movement in a second that, in fact, you can see how it could be driven by this Brownian motion. So this is a very simple peptide. You'll see it starts out as a linear code, and you'll see it jiggle and writhe around, driven totally by Brownian motion, but in 6 microseconds to get into its final structure required for its function. So these have been hard concepts for a lot of biologists to realize, that this linear DNA code codes for that linear protein code, but within it is all the information to get to this stable conformation in microseconds, not in a longer period of time. Also coded into every DNA gene and into the proteins is what's called the N rule of degradation. The N-terminal amino acid determines how stable that protein is and whether it will last a few seconds, a few minutes, or a few hours. And so this coding, we're still learning all the rules of this, but hopefully it's becoming clear that this linear code contains tremendous amounts of information. In fact, when there's slight errors even in protein folding, it leads to different disease states that we're all familiar with: Alzheimer's disease, mad cow disease, this accumulation of protein junk if the proteins can't be processed and broken down well. And I thought one of the most interesting was, because it was quite a while ago now that Lap-Chi Choi and Francis Collins found a single mutation in a chloride ion channel gene associated with cystic fibrosis, and everybody thought it was just a non-functional protein in the membrane, but it turns out this mutation causes the protein to not dissociate from one of the chaperonins. Chaperonins are protein molecules that help the folding of very complex proteins, and so it never gets folded properly, never goes into the membrane, and ends up going out in the trash. When you can't get rid of the protein, it's like garbage accumulating in the streets. It blocks traffic, you can't function normally, and it turns out this is really critical in terms of what happens at night when we sleep. Our mitochondria and other proteins get broken down in the brain, and if you don't have adequate sleep, you don't have time for the trash to be taken out, and that's what your brain feels like in the morning. So I've been told. So all this is a process of dynamic renewal. Looking in human cancer cells, proteins have these relatively short half-lives. We constantly shed and make new skin cells. So each of us shed about 500 million skin cells every day. That dust in your house, that's you. And you wonder why all this dust keeps accumulating. It's because you're there. So you can replace the entire outer skin in 2 to 4 weeks. We have to replace 5 times 10 to the 11th blood cells every day. And even with normal child development, about 50% of all the cells die off in organs as they get properly formed. So every aspect of life is based on cells dying at certain times, proteins having short half-lives and having to be replaced. If you take DNA out of any life form, the cells die. Without this software to be constantly read, to make new proteins, to make new cells, you can't have a life. So we decided to, as I said, go this other direction, and we wanted to rely on historical things, and there's a concept in chemistry of synthesis as proof. And so if you look in the chemical literature every year, there's 10,000 papers or so published with the title somewhere, Proof by Synthesis. This sort of goes back to Waller when he made urea for the first time chemically. And this was considered a big deal because all the vitalists said that molecules from life could only come from life. And this was supposedly the first real hit on vitalist when he showed that you could make these life things chemically. So we wanted to start with the digital code in the computer, see if we could chemically remake the genetic code and recapitulate life from that. And it turns out this was a long and difficult journey because nobody had taken it before, and we were just asking very simple questions. How many genes are essential for life? What's the smallest number of genes necessary to have a self-replicating life form? And ultimately, could we design and construct one? And as soon as we went down that way, we had some new questions. Would chemistry even allow us to make these large DNA molecules? And if we could we find a way to boot them up? So we decided to start small. We started where Sanger left off with the Phi X174 genome. It's only around 5,300 letters. And we chose this for historical importance. Clyde Hutchinson, who's part of our team, actually was a visiting scientist in Sanger's lab when they sequenced the Phi X genome in 1977. And he always walked around with a sample of this virus in his pocket that he took from Sanger's lab. So we had an original, scientists are strange, you know that. And it's also known that the Phi X genome couldn't tolerate many variations. So we thought it would be a good test for the synthesis veracity to see if we could accurately make this genome. There's been machines around for a long time that can make short pieces of 20 or 30 letters of genetic code. But longer pieces have really eluded science. And the machines that make these actually introduce a lot of errors. It's called an N-1 rule. The longer you make the piece of DNA, the more errors there are in it. So we had to come up with new ways to correct these errors. And finally, we decided to go ahead with the synthesis. It took 2 weeks going from the computer code to make this chemical molecule. And the fun stage came next when we injected it in the bacteria E. coli. And what happened, as soon as this synthetic piece of DNA went into the bacterial cells, the bacteria started reading this piece of DNA immediately, started making proteins. The proteins self-assembled to form the virus. And once numerous viral particles were made, the cells burst open and the virus infected other cells. And that's how we detect it. The yellow is a lawn of bacteria, and when you see these clear spots, that's where a virus killed the bacteria. So we call this a situation where the software is building its own hardware. All we did was put in a piece of chemical software, and what we got out of it were these viral particles that could infect other cells and make more copies of themselves. Our goal was not to make a virus, but to make an entire bacterial genome, but we at least reasoned if we could make viral-sized pieces accurately, we could maybe find a way to assemble them all together into the chromosome. So we started down this route. We started first with the smallest genome that was known of around 580,000 base pairs, and we made 101 cassettes that were viral size. And each of these had to be chemically made on the lab bench. It was a very slow and arduous process that took us a few years. And we basically, this looks like a Final Four playoff, but we took pieces and assembled them together chemically. Then we had to grow them at each stage in E. coli, sequence them to make sure we hadn't introduced errors, purified it, and then would go on to do the next step. We had a whole team working on this chemistry. And the problem was when we got up to molecules that were about a quarter of the genome, 144,000 base pairs, the bacteria E. coli didn't like these large synthetic pieces anymore. So we looked around for another system, and being a fond of the products from brewer's yeast, we looked at that very important engine in science because this yeast can take up very large pieces of DNA. In fact, it has a unique property. If the pieces overlap with exact sequence, it can actually assemble them together in a process called homologous recombination. So we found all we had to do was put the 4 quarter molecules into a yeast cell with a small vector that just contains an artificial yeast centromere, and yeast immediately assembled these pieces together. So we isolated it, sequenced it, and that's what we reported in 2008. So at the time, this was the largest molecule of a defined chemical nature ever produced. And it was the completely synthetic bacterial chromosome. And this was a huge step in what could be done with the chemistry. The chemistry continued to improve, mainly by hiring younger scientists. Dan Gibson started as a postdoc, and he was looking at this cumbersome synthesis that we were doing on the lab bench. And he worked out a way that we could combine just all these little pieces of DNA with the 3 key enzymes in one test tube at 50 degrees centigrade, and everything would get automatically assembled. And when he proved this work, he came to Ham-Smith, Clyde, and myself and said, "I don't know what took you old guys so long." And so we call this the Gibson assembly in Dan's honor because it's such a powerful method. And aside from changing the pace of what can go on, it actually allowed us to start to automate the entire process now, because we could then go from the digital code in the computer using a robot to make pieces much more rapidly. So the chemistry was constantly improving, and so we had a second team that was working on how do you boot up a synthetic chromosome. And this was not trivial in any way. In fact, I describe this process in the book, and this paper that we published in Science in 2007 is, I think, on a philosophical level and an information level, probably the most important paper of my scientific career. In this paper, I think, really surprised the entire scientific community, because we showed that we could take the DNA software out of one cell, transplant it into another, and completely convert one cell into the other. And because this is so central to the whole process, synthetic DNA, I'm going to walk you through it. And the 2 scientists here, Carole Letigue was a postdoc from France, and John Glass really did hundreds and hundreds of tedious experiments to work out the details of this. Basically, what we did is we isolated the chromosome, the genome, from one bacterial species called M. mycoides, and we worked out a way to transplant it into what we call recipient cells, M. capricolum, and these are about as far apart as humans are from mice, so about 10% different. When we were working this out, we didn't know if we could just use naked DNA or if there were going to be some key proteins required for transplantation. So we did what Avery did in 1944. We treated the chromosome with proteinases to remove all proteins to see if we could work it out and show that it was just naked DNA. And we had a couple gene cassettes, one so we could select for this chromosome, and another so it would turn cells bright blue if it was transplanted. The techniques are much more tedious, and the book goes into detail on this, but you can't pipette this DNA because it's very brittle. So we have to move it around in tiny gel blocks and then dissolve the gel with enzyme just before the transplantation. So none of this was trivial at any level. And we have this very sophisticated movie to show you what we think happened. So we added the M. mycoides chromosome to the recipient cell. And now we have an unusual situation. We have the complete body and phenotype of one species, and it has 2 sets of genetic software. What we think happened was like with the synthetic DNA with the virus. As soon as we put this chromosome into the cell machinery just saw it as DNA, bound to it, and started reading it, and started making proteins. And some of the earliest proteins produced are the restriction enzymes that Ham-Smith got the Nobel Prize for in 1978. And these enzymes that came from the new chromosome recognized the DNA in the cell as foreign DNA and chewed it up. So now we have the body of one species and the software of another. So what happened? In a very short time, we had these bright blue cells, and when we interrogated these, they only had the DNA that we transplanted, and they didn't have a single molecule or characteristic of the original recipient cell. So simply by changing the software, because it drives the formation of new proteins, and the proteins turn over in the cell, there was not a single molecule left from that original species. So I think this really helped prove that DNA is a life-based software system, and if you change the DNA software, you change the species. So we wanted to do this with the synthetic chromosome to prove the whole loop, starting with the computer, but we had a problem because we were actually doing the final assembly of the bacterial chromosome in a eukaryotic cell, yeast. So we had to find a way to isolate the bacterial chromosome from the yeast cell to transplant it. And so to do this, the team developed ways to actually grow bacterial genomes as eukaryotic genomes. And this was amazingly simple to do. We just simply add the synthetic yeast centromere to a bacterial chromosome, and it behaves like a eukaryotic chromosome. Major steps in evolution can be very simple, and these chromosomes are very stably maintained. So we now had the situation where we had the mycoides chromosome in yeast, a way to isolate it. We isolated it in the transplant, and it didn't work. This little problem took the team 2 years to solve. And the solution, in retrospect, should have been obvious to us, seeing that we had the Nobel laureate who discovered restriction enzymes. The DNA in the mycoides cell is methylated. And that's how cells protect themselves, their genome, from their own restriction enzymes. And so we proved this by purifying the 6 methylases from mycoides cells. And we showed if we methylated the DNA after we isolated it from yeast, we could get transplantation. And we ultimately proved it by removing the restriction enzyme genes from the recipient cell. In that case, we could take naked DNA out of yeast and get transplants. So little setbacks like this can be very costly. If we were not funding this ourselves, it's doubtful that a government study section would have given us 2 more years to keep going. But fortunately, we had it. And it turned out this method ended up being very critical out of this side road discovery, because we've now shown that you can take bacterial genomes from a wide range of bacteria, add this yeast centromere, and grow them as a yeast chromosome. And what that allows is we can use all of yeast genetics to make changes in the bacterial chromosome. Most bacteria cannot be worked on because they don't have genetic systems for making changes in the genome. This opens up now the ability to change genomes very rapidly. We can go through this process, isolate the chromosome, methylate it if necessary, transplant it, and just work around the circle, making lots of changes. So at this stage, we were pretty cocky that we'd solved all the problems, and we decided with the new synthesis techniques to make the much larger M. mycoides genome at 1.1 million letters. And Dan Gibson largely drove this process with his new technique. We started with pieces that were 1,000 letters long. We put 10 of those together to make pieces that were 10,000 letters long. We put 10 of those together to make pieces that were 100,000 letters long. And then we put 11 of those in yeast and assembled the entire mycoides genome. We did the transplant, and it didn't work. And so software engineers, some of you are most likely software engineers, have debugging software to find the errors in your code. We didn't know if we had a design error, synthesis error, a sequence error. And so we had to devise a biological version of debugging software to find out why this chromosome didn't support life. And basically, we made from the native genome pieces that matched the 100,000 letter pieces, and we could substitute them one at a time to see if they could support life. 10 of them did, 1 of them didn't. So we had to go back and resequence that piece. We actually, the first time we sequenced it, used new generation sequencing technology, and we went back to the Lee Hood method to get it right. And so Sanger sequencing is still the gold standard for any forensics or key diagnostics. We found 1 letter deletion out of the 1.1 million, and it was in a key gene, and that 1 letter difference made the difference between life and no life. We corrected that 1 letter, did the transplant again, and got these beautiful blue cells, and there's much more drama described in the book with it, because this was a very long process to get there. We sequenced the DNA, only the synthetic chromosome was in these cells, and every measurement was there were no molecules left as before from the recipient cell. We also added some extra markers to it, so that this was the cover of Science in 2010 when we reported this. We think it's very important when you're making synthetic species to mark them as synthetic. Initially when we did this, we were just using the single amino acid code to try and write words and names in DNA, but that doesn't cover the whole alphabet. So we devised a new code where we can write the entire English language with numbers and punctuation in the genome. And we put 4 major watermarks built into this species. The first watermark contained the ability, the code for translating the rest of it, because it's a pretty radical code from what was used before. It also contains a URL, because this was the first genome to have a computer as a parent. People were instructed if they could read the code, they should send an email to the species saying they had solved it. And a number of people did, some of them quite quickly, in fact. It also contains the name of 46 scientists that contributed to this work over the 10 or so years. And when we published the first genome sequence in 2008, just using the single amino acid code and just putting our names and the names of the institution, I was accused of not being very imaginative, and we were supposed to put in something like a small step for bacteria, a big step for humanity or something, but so I came up with 3 quotations from the literature that I thought were more apt. The first is from James Joyce, to live, to err, to fall, to triumph, to recreate life out of life. That somehow seemed to fit. The second is from Oppenheimer's biography, American Prometheus, see things not as they are, but as they might be. And the third was from Richard Feynman, what I cannot build, I cannot understand. After a few people solved this code, we posted it. It got a lot of attention, and the first major response we got was a letter from James Joyce's state attorney wanting to know if we had permission to use the quotation. James Joyce was dead. I mean, we're good, but we're not that good. And under American law, you can use up to a paragraph without permission with proper attribution, so we dismissed that. Then we started getting email from a Caltech scientist saying we had misquoted Feynman. But if you look on the internet, this is the quotation that you find over and over and over again. And it turns out one of his biographers made a mistake. I think it was him. And the proof that it was wrong, they sent a picture from the Caltech archives of Feynman's blackboard. And the proper quote, which is actually a much better quote, is, what I cannot create, I do not understand. And I think that's very, probably the most appropriate quote for this whole approach to biology. So we've gone back and corrected the genome so Feynman will rest peacefully and going forward. But watermarking is an important part of things. We're starting whole new branches of life. They're going to be the synthetic branch. And if they're not clearly watermarked as synthetic, it could really confuse evolutionary biologists when there's unique combinations that couldn't occur naturally. But we actually have software at Synthetic Genomics, computer software for designing new biological software, and we're trying to organize genes and life into a more functional format. We call it defragging the genomes because evolution's been pretty messy over 3.5 billion years, and nothing's, if it's in a logical order, we don't understand the logic. So we're improving on the work and reorganizing things. And we're using these in a number of different areas. I'll just give you a couple of examples, and there's many more in the book. Earlier this year, the very first genome-based vaccine came out. This is work that we did with Rino Rapalli over a long period of time based on sequencing the meningitis genome in 1996 and picking very stable genes that were highly conserved as vaccine targets. And now this new vaccine works against all strains of meningitis B. But it still took 17 years from our starting point to getting something on the market. We were looking how to change this process. And we decided to, because a flu vaccine has to be remade each year, sometimes more than once if there's new pandemics, that this would be key for our new techniques of being able to write the genetic code. And just a reminder, in 1918, flu killed about 3% of the world's population, and it's only minor variants in the genome that yield these new strains. So a lady that was killed by this virus in 1918 was buried in the permafrost. Her body was recovered, and the virus was still recovered from her body and sequenced, and shown it was only a few letters of genetic code different from some of the modern circulating strains, which is why people are so concerned about dangerous flu reemerging. The Venter Institute is a key part of the World Surveillance Network, where we're actually sequencing new strains as they emerge around the world, so that we have the digital information very rapidly. And then we're using our new synthesis capabilities to immediately make new strains. And this just got a real-world test with the outbreak of H7N9 in China, and starting just in April of this year with 33 cases and 9 confirmed deaths. And the problem is this doesn't show up as a disease in birds, so it's very hard to detect its spread. But there is now clearly human-to-human transmission, and more cases have been emerging, and because of the high lethality rate, a vaccine was badly needed. The Chinese did the right thing. They sequenced the DNA right away and posted that on the internet, but would not send the virus out of China, and you don't necessarily want this going around in the mail anyway. So at the request of the government, we synthesized this in a matter of hours, and our synthetic vaccine, our synthetic virus was the only source that the CDC and BARDA and Novartis had to begin to understand the biology of this virus and to start work on a vaccine. Until this year, and even for production for this year's flu vaccine, for the US it uses around 900 million chicken eggs. This is very ancient technology, but it's very tough to change manufacturing processes with the FDA. But Novartis, with the help of BARDA and the government, built this giant facility in North Carolina that uses mammalian cells now for producing the flu vaccine. So we wanted to go one step further, and this is part of the key interface between DNA and the digital world and going the other way. We're actually building what I call a biological teleporter, where we can actually send something through the internet, such as a virus sequence, and we have a robot that we call a digital biological converter that converts that digital signal back into biology. And we have the prototype of this. We have a more manual version working at that Novartis facility. And so with a new outbreak, instead of trying to send physical viral samples across country or around the world, we can send it through the internet in less than a second. And our process will, within 8 to 10 hours, produce a new vaccine that can go into their production system. So got us thinking a little bit more about biological teleportation. And as an example, and it may be an important one, there's been a problem ongoing for decades of how do you get samples back from Mars? And to get them back from Mars, you have to send a rocket up there that's big enough to blast off from Mars and get back. And some of the ones bringing samples back from asteroids or sampling have crashed in the ocean, and that's not particularly good if you're, according to the Alien Space Act, where you're not allowed to bring in alien species without extensive quarantine. And those of you who watch the ones that are old enough to watch the astronauts returning from the moon, after they got out of the water, they put them in these mobile homes for a couple weeks to make sure they weren't contaminated. they were already swimming in the ocean, so I wasn't clear how that was going to help, but I think it was more for public relations. But we worked out if we just send it as electromagnetic wave, we can send biology back in as little as 4.3 minutes. And instead of it landing in the ocean, we can, in a secure life, rebuild that Martian life in a lab here. While this is illustrative of what can happen, we think it's going to be used in the future going in both directions. But you know, this is the earliest stage of getting biology out of the internet. Right now, we can download proteins such as insulin from the internet. We can download viruses, phage that kill specific bacteria, and we can even do these simple self-replicating bacterial cells now. We're thinking of hiring some 11 to 13-year-olds to tell us what the applications really need to be going forward. But when you think that as soon as you can get from the digital world back in the DNA world, you can, in theory, reproduce all of life. So even thinking of this for vaccines, instead of maybe just having one site in North Carolina at the Novartis plant, what if every country had one? Because even though we had flu vaccine in this country with H1N1 pandemic, the vaccine was only available 2 months after the pandemic peaked, and it was only available for healthcare workers and senior government officials for a long time. A lot of countries it took a year or so before they could get the vaccine. So we can send a new vaccine around the world in less than a second as a digital wave. And if you have one of these converter boxes, maybe it's in each town, each city, or ultimately in each home, it could be the end of pandemics. Because unlike the movie Contagion or unlike previous pandemics, if you have a vaccine, pandemics get stopped in their tracks. And I think these are just the earliest applications that we can think of that have real practical applications right now of converting life into digital code and converting it back into life somewhere else that we hope will stimulate people's imaginations. In 2010, when we announced the first synthetic life form, President Obama asked his new bioethics commission to take this on as their #1 challenge, and they issued this report in 2010 that you can download from the White House website, and it's a very sophisticated report. They interviewed all the players on the pros and cons and the worriers and the warriors, and came up with a very balanced report of the excitement of how this can change all of manufacturing going forward, but there's downsides of risk of bioterrorism, and came up with a really balanced way to proceed. And in Life at the Speed of Light, I talk about this whole history back when we started with the first bioethical review before we did the first experiment, and that makes this pretty unusual as a field of science. So I'm delighted to take your questions, and thank you for your rapt attention.
All right, folks, we have about 15 minutes for questions. So once again, please come to either one of these 2 microphones, form lines down the aisles, and please do keep your question in the form of a question. Thank you.
Hi. I was wondering if you could speak about just a little bit about the role of software simulation in the work that you're doing, and if you see it growing. And is it gated by computer speed, or is it gated by sort of knowledge of what's going on at the sort of atomic level that you simulate?
No, it's a good question, and actually devote quite a bit of space in the book to talk about the computer simulations, because most of them are based on these early genomes that we did in the mycoplasmas. And for the last 20 years, people are getting better and better at first modeling pathways like glycolysis, to this last year, a team at Stanford put a complete computer model together of this mycoplasma cell that we made using a very large cluster of computers and 127 different algorithms. And so I think it's gone further than anybody could go before, but you sort of answered your question at the end. It's based on the biological knowledge. You can't model something that we don't know or understand. And the problem, even with the minimal genome we're building now, about 10% of the genes are of unknown function. We just know if they're not in the genome, you don't get life. So my uncle's sitting right behind you. He designed some of the Boeing aircrafts we fly in. Imagine doing that design without knowing what 10% of the parts actually did. Or maybe that's what happened. I don't know.
Thank you.
Okay.
This is actually kind of a follow-up on that, or your answer to it. So there's a lot of DNA that doesn't seem to code for anything that we're aware of. Do you have any sense of what that might be doing, or is it just junk DNA?
Well, in bacterial genomes, they're actually wired pretty tightly. In viral genomes are even more condensed, where you actually have overlapping genes on a single strand of DNA. People arrogantly called all the parts of the human genome that not code for proteins as junk DNA, which was kind of a sad state of genetics, and both Lee and I constantly fought others about that. And that's where all the discoveries are happening. And first the microRNAs were discovered, all these regulatory RNAs that don't code for proteins, but regulate gene expression. Our physiology is pretty complex. And even the parts of the chromosomes that control how they open and how they're read for making RNAs is a complete part of things that we only have minimal understanding of. So I know some people I'd like to remove their junk DNA from, but I don't recommend anybody do it. Yes?
So as you try to move from synthesizing single cells to multicellular organisms, what do you see as the stumbling blocks, and how might you overcome them?
Yeah. So all species are based on what was described in the 1800s as cellular-based life. And if there's something common to all life other than DNA, it's the cellular structure. And so multicellular species can be loose aggregates of cells and biofilms, or they can be much more structured like we are. I've argued for a long time that the two most important things to understand about human body is understanding how stem cells work and understanding how the genome codes for these different changes. Because each stem cell has the same genetic code in it, but just with a few hormone tricks, you can get something to make neurons or heart cells. So we have to understand that coding a lot better. And again, it's pretty hard to design something without knowing those principles. So a key part about the future of this field right now, it's based on empiricism. And we don't like to confess that, but biology is still a very empirical field. The government review process doesn't want to recognize that. If you don't know the answer to the question you're posing, you can't get funds for it. So what does the empiricism mean? It means we have to have large numbers. So we're setting it for DNA synthesis, where we're working on a robot where it can make 10,000 genomes a day and then score those for the functions you're looking for. It's simple, did it live or did it not live? But if you're looking for more complex traits, you can do that. And a self-learning system we can learn thousands of times faster than a scientist. But it's still based on large numbers because of all the things that we have to discover empirically. So that's a long way of saying it's going to be tough to design multicellular organisms, but we're working on eukaryotic systems and others that, in theory, can easily go in that direction.
Thank you.
Yes?
Actually, when you were talking about the NHS funding, I became a bit interested in actually maybe personal and professional development, if you talk about that. Confidence is something Lee brought up, but and so clearly that's an example of that, right? 2 years, maybe plus. But you also seem really humble here. I mean, you're sort of talking about the junk DNA. I guess I'm curious what gives you that confidence. What if if you could answer that question maybe by taking a bit of a detour on your past, where you maybe didn't have confidence, maybe that I've heard about a long cold swim, for instance,
Yeah.
And suicidal ideation, and I think that's
Well, that was the
very
previous
similar.
book, so, but look, it's like reading this book from this Frenchman, Francis Detect, who was totally confident in 1901 that based on life having mechanistic basis, that at some stage it should be able to be replicated through science. I think all the things that we have done, there was no question to me that they would work. It was a question of how long it would take and what route it would take to get there. And if you have the wrong belief system, you can get a lot of trouble with it. But I think mine has been based on a combination of intuition and paying attention to what the data's saying. It wasn't a blind belief system. And so our journey to make this first synthetic cell is one that could only happen, I think, today because of independent funding versus trying to convince a study section. And we even had some nervous board members when each year we had to say it didn't work this year, but we think it will next. And that story kept going for 10 years, but we had very steady progress along the way. And there was no question that it would eventually work. There was a question whether we could get it to work. But I think these principles of biology that I try to cover in this book are pretty fundamental principles of how life works. And if they're true, which they seem to be supported by the data, all the things we're proposing going forward from here are pretty logical extensions. It's hard to predict the exact time course and when they'll occur, but I don't think there's anything super magical about these steps. But Lee's made a lot of breakthroughs, and you spend a lot of your career trying to convince people to change. And but that's been the history of science. Nothing convinces people like experimental data, though. In the arguments I like to show, showing the data and showing the results, and you have a living, self-replicating synthetic cell, it wins a lot of arguments. Yes, sir.
Now that you've created a living and self-replicating cell, what do you think the next biggest step is in genetics and in producing genetic material?
Well, the next biggest step that we're trying for is actually going the other way. We're trying to make the smallest genome of a living, self-replicating organism to understand what those components are. Because if you want to design things, and so one of the scientists in the 1800s I talk about in the book said, give me a protoplasm, and they didn't even know what protoplasm was. They didn't know what proteins were. But give me a basic protoplasm, and I can recapitulate the entire animal kingdom. And I think that's true in theory if we can start with a minimal cell with a set of genes responsible for all these steps of DNA replication, cell division, et cetera, energy modules, that we should be able to add components to that minimal cell and recapitulate evolution and even change evolution and make it go thousands of times faster. But we want to get down where we understand the basic components, or we can never do it in a priori logical design fashion. You can only do it empirically. Yes, sir.
Okay. So the basic thrust of your research and accomplishments is to distill the information from DNA and then be able to inject that into a new cell. And so then that cell then takes on the information of the synthetic DNA with quotes from James Joyce and so forth and replicates. So, and in the last part of your talk, you were talking about being able to do that in a kind of exobiology sense of a probe to Mars. You find something that looks like life. Presumably you have a little machine that sequences DNA, send it back to Earth. So my question is, the other half
So far, you showed a really good understanding of what I said. So.
Okay, thanks. Okay. So my question is, you have that information, but do you know, I mean, I guess it's an assumption that if you insert the DNA information, you have to insert it into something. It's a cell. The cell takes that information and replicates and, you know, has all this stuff going on. There's a second half of this thing, which is the cell without the genetic information. Is the assumption, I mean, it seems like I guess it's a refutable assumption, but that all life everywhere, so I mean there's life on, you know, someplace besides Earth, has the same kind of machinery that takes the information from the DNA to produce life? Or, I mean, it could be that...
That is clearly the assumption, and I spend almost a whole chapter talking about why I think that's so in the book. You find the same chemicals throughout the universe.
Right.
You find the same basic molecules of amino acids and nucleic acid bases on this planet, basically everywhere anybody looks.
Right.
And we exchange about 100 kilograms of material between Earth and Mars every year.
Right.
In fact, statistically, you can't take a shovel of dirt on this planet without having Martian soil in it, and vice versa. So, and the data shows that Mars was covered at least twice with water, and that you go down a kilometer or so and you find a lot of ice, and below that is probably liquid water. Clearly, we introduced life on Mars, or Mars introduced life on Earth. We don't know which way it went. If we find life on Mars in these frozen layers or in the liquid water, I doubt that we'll find it on the surface. I would be extremely surprised if it didn't match very closely something on this planet. And I talk about the reasons for that. Even the experiments we've done here, Lee talked about how we sequenced microbes around the globe, but we also sequenced deep in the Earth. And a remarkable difference was a mile deep in the Earth, we found the same density of life that we find in the oceans, but you don't find the same diversity, because the two major drivers for evolution are sunlight and oxygen. Deep in the Earth, there's no sunlight, and there's definitely no oxygen. And the closest relatives, and they were remarkably close to what came out of a coal bed methane well in Colorado, it came out of a volcano in Italy. And I'm not suggesting all this is interconnected in the deep surface, but you know, when the estimations are meteors probably removed oceans on this planet once or twice. We had these deep reservoirs for life, and they have been conserved over billions of years. So I think it would be a far bigger scientific discovery if there ends up being no life whatever on Mars and no trace of it, and if it was substantially different. I have an open mind to it, I just don't expect it. Yes, sir.
So you talked about taking the genome from one bacteria cell, putting it in another bacteria cell, and it kind of booting up almost automatically. But you're also producing this bacterial genome in another cell, in a yeast cell. And I was just wondering, do you see any kind of rebooting within the yeast cell when it has the bacterial genome in it? And then also just, what's your sense for how far along kind of the species continuum you can go, taking one genome from one species to a...
No, those are both very good questions. So the genetic code in the mycoplasmas is slightly different than it is in all other species. In one, the triplet codes for a tryptophan, and the other species it codes for a period at the end of the genome sense, a stop codon. So yeast would read it as a stop codon and probably not make functional proteins. We didn't want them to be expressed in yeast, so we were happy with this. But we are actually changing the genetic code so that can happen as well. And we're trying to, and I talk about this a little bit in the book, we're trying to design a universal recipient that has the protein robots and the tRNAs that can read basically any genetic code we put in and boot it up. I think it is that universal, with the exceptions for these subtle differences in the genetic code. I was told we have to stop. We're down to one over here. Is it a speech or a quick question?
Quick question.
Okay.
First of all, thank you very much for all your work and hope to see more from you in the future. It's really been exciting. The bioteleporter thing really caught my interest. And what do you think it's 20, 50, 100 years out before we can print out a squirrel, or we're going to print out you on Mars or something? What are you hoping for?
I have no desire to print a squirrel, but you know, if that works for you, then you know, it's. I talk about quantum teleportation in the book because it's something that could really change teleportation, but it's not like Star Trek of Beam Me Up, Scotty. It's in one of the founders of quantum teleportation described it as it's a lot easier to send the instructions for making a 747 through the internet than it is to send you a 747. And so, yes, squirrel genomes and human genomes can be sent through the internet and sometime in the future replicated, but you're starting with a very young squirrel or a very young human that has to go through all of human development. We will not be sending you from the internet and getting you back immediately on the other end, although I somewhat joke about this. My genome has been now being broadcast from Earth for 14 years. And I tell people not to be surprised when a lot of me come back and invade. So anything's possible. Yeah. Real quick, both of you, and then we'll quit.
It's on a practical note, and I found your presentation fascinating. On a practical note, I've read there are attempts to introduce a DNA of mammoths into a modern elephant's egg and recreate a mammoth, which is a wonderful way of trying to recapture extinct or declining species. Are you related to the research project?
No.
With mammoths?
In fact, I think these are press-created myths that the mammoth genome sequence and the Neanderthal sequence that one of our colleagues would like to impregnate a lady with is so faulty and so fragmented that you couldn't get anything out of it. And these are, you know, we all know that everybody likes to jump to the sensational stage of things. I have no need for a woolly mammoth. Maybe somebody does. And Washington's full of Neanderthals. So. Last question. I'm sure it will be immensely profound.
Oh, thank you, sir. Just wanted to say, again, thank you very much for your talk. And there's a lot here you've talked about tonight that's just truly miraculous, particularly the case of vaccines. I mean, the chance for a positive impact on billions of people, that's so fantastic. But of course, technology can also be used for nefarious purposes. And so I guess I'm tempted to ask you what perhaps scares you about the possibilities of this technology. But what I'd prefer to ask is, I guess, what do you think is necessary? I mean, you have the White House report to have reasonable protections in place, but not interfere with the work of scientists who are doing good work like yourself.
So it is a good question, and the White House report does address that as a great starting point. I would point to the last 40 years of success of the scientific community of self-regulating. Basically, millions, if not tens of millions of experiments have been done of putting genes from every species into E. coli without the monsters appearing on the doorstep. And the reason for that is the laboratory E. coli with other genes can't grow outside of the laboratory. And these are some of the things the Obama Bioethics Commission talks about, is developing lots of approaches for kill switches, et cetera, in any organism that's set up for industrial purposes, so it can't live outside of the system. We do need intelligent protection. On the downside, our biggest threat is actually new emerging infections. It's not somebody synthesizing a genome to try and kill people. But guess what? The solution is the same for both. We need new antibiotics. We need new antivirals at a time when the pharmaceutical industry is not interested in making these because they don't make enough profit off of individual antibiotics. They tried to make a few antibiotics that worked against all microbes, and guess what? We got resistance. Now there's more people dying a year in the US from bacterial infections than from car accidents. So it's a very dangerous period in history. If we don't change that pretty quickly, we could end up in the equivalent of the pre-penicillin era of medicine. And I don't think we want to go there. So if we have these new agents that work against new emerging infections, we'll certainly be covered if somebody wants to use this for more nefarious purposes. But thank you for the question. Thank you.